Hi guys, this video is going to show you how we set up our Bio CRISPR Lab. Remember, the goal of this lab is to disable or turn off the LAC-Z gene. When the LAC-Z is functioning, it makes the bacteria appear blue. But at the end of this lab, we want to see bacteria that are white, which means the gene is not working. To disable the gene, we're using CRISPR, which is a gene editing tool naturally found in the bacteria already. So now, here's how we set it up. Before starting the lab, bacteria was grown onto starter plates. Both of these starter plates have X, which is an antibiotic, and one of them has arabinose, which is a sugar. The DNA repair machinery of the bacteria needs the arabinose in order to function. Here's what the starter plates look like when we start the lab. As you can see, the bacteria on the starter plates is blue because the LAC-Z gene is functioning. Now let's look at the other materials that we need to do this lab. We've got yellow inoculation loops, pipettes, new test tubes, some micropipettes, and our samples, as well as our starter plates. PDG is the donor gene with guide RNA. PD is just the donor gene, no guide RNA. And LB is LB broth, which is basically food for the bacteria. We also have a cup of ice, which you'll see in a moment, and in there is our fourth sample, which is transformation solution. It has to be kept cold. First real step of this lab is labeling all of our new test tubes A, B, C, and D. Once labeled, we place the tubes on ice. Then add 250 microliters of super cold transformation solution, which is our TS sample, into each of the test tubes. After adding the transformation solution, we put them back on ice right away. Now we need some bacteria, and we're going to get them off of our starter plates. First, we're using the X plate. We're going to scrape 5 colonies, which are little dots of bacteria, off the plate and put them in test tube A with the transformation solution. It's important to really swirl this around for a while, probably about a minute, in order to get all of the bacteria off of the inoculation loop and into the test tube with the transformation solution. After using your inoculation loop, you take it to the biohazard trash. Now, just repeating the same process for test tube B, again using about 5 colonies of bacteria from the X plate. Now for test tube C and D, we'll use bacteria from the X ARA starter plate, so the other one. Just making sure, again, that all of the bacteria gets into the transformation solution, swirling around for about a minute, and then taking used inoculation loops to the biohazard trash can to prevent any cross-contamination. Now it's time to insert new DNA into our test tubes. To do this, we're going to use a micropipette because we only need 10 microliters. We're going to start with the plasmid donor. It was hard to get our fluid out of these, so we're going to centrifuge them. This is the centrifuge. It will push all the fluid to the bottom. When you use a centrifuge, it's important to balance out your samples. So I placed 1 test tube on either side of the centrifuge and then turned it on. It just spins the samples really fast. Then once I take them out, all of the fluid will be pushed to the bottom and I'll be able to actually get it out of my test tube. So now back at the lab station, we're gonna put 10 microliters of our plasmid donor into test tube A. Once we have it all in there, we remove the pipette tip, discard it, and then give the test tube a couple flicks just to make sure that everything's suspended and mixed correctly. We're also gonna add the plasmid donor gene to test tube C and repeat the same process with that test tube. For test tubes B and D, we'll repeat the same process as before, but instead of using PD, we're going to use PDG for these. So these samples are getting the donor gene and the guide RNA in test tubes B and D. Again, just making sure that you give it a couple of flicks to suspend all of the bacteria and the solutions, and then returning them to ice once you have finished. The next step is to incubate on ice for 10 minutes. So we just sit back and wait for that amount of time. After 10 minutes, we're ready to move on to the next step of the lab. The next step is to heat shock our bacteria in a hot water bath. We'll put them into this little styrofoam floaty so that they don't sink to the bottom of the water bath, and then take them over there. The point of this step is to try to get the bacteria to open their cell walls and accept our DNA. The bacteria need to sit in the hot water bath for exactly 50 seconds, and as soon as the 50 seconds is over, we'll return them back to the ice. Once we get back to the lab station, the bacteria are back on ice and they incubate for 2 more minutes. Once the time is up, we can move on to the next step of the lab. From here, we're going to use some pipettes to put 250 microliters of our LB broth into each of the test tubes. Each test tube is going to get the same LB broth and the same amount of LB broth. This is basically the food for the bacteria, so they'll have something to use for energy while they grow. Just like you saw before, after adding the liquid LB broth, the test tubes will get flicked a couple of times just to resuspend the bacteria and to make sure that everything's mixing up really well. Once every test tube has LB broth, it's time for a 20-minute incubation. Now time is up and it's finally time to plate our bacteria. We're going to place each of the test tubes onto a plate that corresponds with their letter. So test tube A onto plate A. We're adding 100 microliters of each bacteria solution onto the plate that corresponds with them, and then moving the bacteria around using an inoculation loop. Of course, we're using a fresh inoculation loop and a fresh pipette tip for each of our different test tubes, and then keeping the plates closed as much as we can to prevent any cross-contamination on the plates. You'll also notice that once again, we're flicking the test tubes before we pipette any solution out of them, just to resuspend the bacteria after such a long incubation time. We want to make sure that we actually get bacteria on the plates so that something will grow. Once we have bacteria on the plates, we just stack them up upside down and tape them together in a stack. Storing them upside down prevents condensation so that we'll be able to see our results. Finally, we just take them to the incubator, which is set to 20 degrees Celsius, which is the perfect temperature for the bacteria to grow. We'll leave them here for a couple days, and then we'll be back to check the results after our bacteria have had a chance to grow. And that's it for our CRISPR Lab setup. Thanks for watching, and we'll see you back for part 2.